Hi, I am Dr. Arsalan and today we are going to study the endospores in bacteria. These bacterial endospores were discovered by Ferdinand Kohn in 1876. The endospores may be defined as these are dormant, tough, resistant, non-reproductive structures produced by the gram-positive bacteria in harsh conditions. When there are harsh or unfavorable conditions to which the bacteria is exposed, then these bacteria produce endospores. These endospores are produced inside the cell of the bacterium, and these are highly resistant, tough and dormant structures which, when favorable conditions are attained, these germinate into new bacterial cell. So the endospores are aimed for the propagation of generation of the bacteria. The endospores may remain dormant for millions of years until the favorable conditions are achieved. These are tough or hard. These are resistant to the harsh conditions, harsh environment, unavailability of the nutrients. These can survive in these conditions. These are non-reproductive structures because these yield only one bacterium per spore, and these are most often produced by the gram-positive bacteria. This is the rough diagrammatic structure of the endospore. The most interior, first of all, this is the DNA molecule or genome of the bacteria. These are ribosomes, the small granules, and these are the proteins Dipicolinic acid. Dipicolinic acid are the proteins which help in dormancy of the endospores. Due to such proteins Dipicolinic acid, the endospores remain dormant for years. This is the cell wall of spore. Then this is cortex. This is the inner wall of the core. This is outer wall of core. And the outermost layer is called exosporium. So these three layers, these are basically called protective layers: the inner core, outer core and exosporium. When these endospores germinate, these layers break up and the endospore is released. This endospore then later on germinate into new bacteria. The endospores are basically composed of small amount of water with cytoplasm and little amount of DNA along with the ribosomes. These are all enclosed in the cell membrane, cell wall, external protective layers of the core protective layers, as you have seen in the structure of the endospores. Next is the position. As we know that the endospores are produced within the cell of the bacteria, so inside the bacterial cell, when the endospores are produced in the center, or these may be produced at the side of the cell. So there are two kinds of the endospores produced in the bacterial cell on the basis of the position. This one is called central endospore and this one is called peripheral endospore. So there are two kinds of endospores on the basis of position: the central endospore or peripheral endospore. Next are the conditions. So endospores are produced only in the harsh conditions. When the conditions are not favorable for bacterial survival, these produce endospores so that the generation of the bacteria may propagate in a proper way. Next is the life of endospores. These endospores remain dormant or inactive due to a special kind of proteins called dipicolinic acid, as we have shown in the structure of endospores, the dipicolinic acid proteins. So the endospores, being dormant and tough structures, these can survive for thousands or millions of the years. On attaining the favorable conditions, these endospores germinate and form the vegetative cell. We were talking about the life of endospores. The archaeologists have revealed strange discoveries, and they found the endospores in Egyptian mummies as well as Minnesota elk lake. Minnesota elk lake is present in the USA, and the spores were discovered in Minnesota elk lake in the fossils, and these revealed the existence of about 7,518 years ago, and the endospores were found still dormant. And when these were provided with the favorable condition, these endospores germinated into the new vegetative cells or the bacterium. Next is the sporulation. Sporulation is the process of formation of endospores. Process of formation of endospores in the bacteria. So this process is particularly called as sporulation. Now the question arises here that whether these endospores are vegetative or reproductive structures. So it should be kept in mind that these are not reproductive structures, rather these are vegetative structures, because endospore give rise to single vegetative cell or the single bacterium. The number of bacterial cells or the vegetative cells do not increase. Therefore these endospores are termed as vegetative structures, not reproductive structures. Or we can say the single endospore gives rise to a single bacterium or single vegetative cell, so there is no increase in number of cells. Therefore we termed it as vegetative structures. Now we'll look into the process of germination of the endospores. So it's a hard and fast rule for the bacteria that the endospores germinate only when the favorable conditions are attained. So during the favorable conditions, the protective layers break. As we have shown in the diagram that the inner layer, outer layer and exosporium, these were three protective layers of the endospores. These break apart. When these layers are broken out, then the endospore is released. This endospore, it uptakes water and it is engorged with the water or swells up. After swelling or gaining rigidity, this bursts up. The endospores burst up and releasing the vegetative cell. The vegetative cell here is called the bacterium. So this is the simple mechanism of germination of the endospores in bacteria. Finally, the examples of bacteria which can produce endospores, notorious for the production of endospores, these are Clostridium and Bacillus. The Clostridium and Bacillus both are gram-positive bacteria, and these are spore-forming bacteria. Particularly, these form the endospores. So these were some important and precious information regarding the endospore formation in the bacteria. Hope you have learned a lot in this lecture. Thank you.